Welcome to the next lecture on human genetics. Please don't distribute. So you may be asking yourself why we are doing a lecture on human genetics within a genomics class. And the reason is that in the field of human genetics, a lot of the focus previously has been on identifying genetic variants associated with different complex traits, characterizing the heritability of different complex traits, and in general, interest in, for example, predicting complex trait phenotypes from genetics alone. What none of those previous studies were able to do, though, is actually give some kind of mechanistic insight into how those genetic variants work. Right? So you can identify hundreds of genetic loci, for example, associated with height. But one of the most important questions is how do those genetic variants that, even if you find a causal genetic variant for height, how does it work mechanistically? And so to answer that question, there's increasing interest in studying the genetics of molecular traits. And so identifying genetic variants that, for example, are associated with changes in gene expression measurements of different target genes. People now look at genetic variants that alter chromosome accessibility, histone modifications, or DNA methylation. And so it's important to then kind of have some background about how do human genetic studies generally work, and how are they applied towards molecular traits. And so the learning objectives for today are basically to understand the advantages of genome-wide association studies, which kind of form the basis of a lot of the studies of molecular traits, and how does that compare to kind of classic linkage analysis. We're going to discuss when you would use, for example, Fisher's exact test versus line fitting to study association with either discrete phenotypes or continuous phenotypes like height, for example. One of the biggest challenges historically with these kind of genome-wide association studies is dealing with what are called confounding factors. And so we'll discuss briefly what those are and what are some of the basic strategies for correcting for them. We'll also discuss a little bit about how to identify population structure and families from genetic data. So population structure is one of the biggest confounding factors of most genome-wide association studies. We'll basically look at QQ plots, which are one way that people kind of diagnose uncorrected confounding factors in genetic studies. And then we'll discuss briefly what the differences between effect size and significance of statistical tests associated with genetic associations are. And so we previously discussed a little bit about p-values and effect sizes. And so we'll discuss it in the context of genetics here. We'll also touch on the topic of fine mapping, which is basically related to the problem that genome-wide association studies identify regions of the genome that are associated with different complex traits, but they don't give you the exact causal variant most of the time. And so fine mapping is the process of identifying the precise causal variants that are implicated by genome-wide association studies. And we'll talk about how, for example, epigenomics helps identify or fine map variants. And then we'll finish off by talking about epistasis and missing heritability. And so the key question that human genetics tries to answer in the context of this class is basically the question of where are the genetic differences that contribute to heritable diseases? And so this class in particular is going to focus on single nucleotide polymorphisms, or SNPs, or more generally, we'll be talking about single nucleotide variants, or SNVs. And so those are basically variants where you just see single base pair changes at a given position in the genome. But it's worth noting that there's a lot of other kind of genetic variation that exists in the human population, for example. And so you can see a lot of like small indels or even larger scale copy number changes in different regions of the genome, but we'll more or less ignore those kinds of genetic changes, mainly because a lot of these association analyses work much better for single point mutation changes. And so it's also worth noting that even though one of the reasons why there's historically a lot of focus on SNPs and SNVs is that they are predominantly the most prevalent kind of genetic genetic variant in the human genome. But it's worth noting that there is a large number of variants of basically larger size. And so this is basically just a plot of the frequency of variants of different sizes in the human genome as estimated by the 1000 Genomes Project. And you can see that even though there's a little peak there at SNPs, there is basically a lot of variants at other sizes in the human genome. And so it's also worth noting that the set of variants that you find in the human population differs substantially depending on which ethnicity that you're talking about. And so this is not a PopGen class, so you're going to have to take a PopGen class in order to really get a good grasp of what we're talking about here. But basically, different ethnicities in the human population have different effective ages. And so what that means is that, for example, in the African population, the number of genetic variants observed is much higher than, for example, Europeans. And so this slide is basically just to say that when we talk about identifying genetic variants associated with different complex traits, there can be a lot of heterogeneity between different populations in terms of, not all variants affect all populations equally. And that kind of makes sense. If Africans have many more genetic variants than, say, Europeans, then there could be a lot of potential for there to be African-specific variants that drive phenotypic variation that doesn't exist in Europeans because they don't have that variant. And so this lecture will focus specifically on genome-wide association studies, which basically involve association of genetic variants to phenotypes using mostly unrelated individuals. And so that's to contrast what you've probably classically learned about, which is association through what's called linkage analysis or family-based linkage analysis. And so here, generally speaking, the idea is that you're generally studying the inheritance of different phenotypes in family pedigrees. And so generally speaking, you have access to a pedigree like one shown here, and you have access to at least different markers or, for example, SNPs. And the idea of linkage analysis, broadly speaking, is that you are looking for co-segregation of both a genetic marker and, for example, disease status through a pedigree. And the stronger that association is, or co-occurrence of a genetic variant with a phenotype, then essentially the stronger the link is between that particular genetic variant and that phenotype.